Good afternoon. I'm Jerry Shadle, a professor at the Salk Institute and director of the San Diego Nathan Schock Center. I want to thank the AGE organizers for allowing this session where we can highlight some of the activities of our new Nathan Schock Center for the Basic Biology of Aging research. As the name indicates, the San Diego Center is actually a collaboration between three research institutes on the La Jolla Research Mesa, the Salk Institute for Biological Studies, the Sanford Burnham Prebys Molecular Discovery Institute, and the University of California, San Diego. As you've heard already from the other Schock Center directors so far, each Nathan Schock Center has a theme, and the theme of the San Diego Center is to tackle the heterogeneity of aging, which is a problem in the aging field that I think needs to be addressed systematically in order to be able to intervene successfully to increase the health span of people. I don't need to really illuminate for this crowd what is meant by the heterogeneity of aging, but just by way of introduction, I think everybody is well appreciates that there is not a direct relationship between our chronological age and what we now refer to as our biological age. And what that means is that individuals of specific age types, say 60 years old, for example, don't necessarily show identical age-related phenotypes. That is, you can have individuals who are relatively fit 60-year-olds and those individuals who are already succumbing to age-related declines and perhaps other types of age-related pathology. And so there's this heterogeneity at each individual chronological age in aging phenotypes that we simply don't understand at this point. Of course, this heterogeneity increases as we age. So in a relatively young population shown here, you can see that most people are relatively fit, and maybe a few people are starting to show signs of aging, but those signs of aging are probably different in different individuals. Not probably, it is for sure. But if you look at an older population, you can really begin to see that there's a marked heterogeneity in age-related phenotypes, where many people are still remaining quite fit, whereas others are becoming frail, for example, or again, succumbing to age-related diseases. And we really don't understand this, but we know that it's an intersection between intrinsic factors like our inherited genetics, intersecting with extrinsic factors such as our lifestyle and environment that are driving these differences in aging trajectory. Even if you can subtract out, to the best of our ability, genetics and environment, there's still an intrinsic level of heterogeneity in aging, and that's demonstrated here with this experiment from C. elegans, the roundworm, just showing a typical lifespan curve of this model organism. You can see that individuals are dying at different rates. So some individuals are dying early and some individuals are dying late. And even if you give them a health span extending or lifespan extending regimen, you can shift that curve to the right, but you really don't eliminate the majority of this heterogeneity. That is, there are still individuals who die early and die late, despite the fact that they have, to the best of our knowledge, identical genetics, and we're controlling the environment as best we can. So there are intrinsic factors that contribute to aging that, again, we don't understand very well. So our premise at the San Diego Nathan Schock Center is that by understanding the heterogeneity of aging, there could be quite a number of benefits to the aging field and to society in general. First, in terms of basic biology of aging research, it will allow us to improve the design, execution, and interpretation of the studies we're doing right now. Second, we believe it'll help deconvolute these complex relationships between intrinsic and environmental impacts on aging. We feel that there's an opportunity for identifying new biomarkers of aging and age-related pathology. And if we can really understand this at a deep level, we should be able at some point to be able to look at individual susceptibilities to specific age-related declines and diseases and be able to maybe even predict how different individuals will respond to different health span interventions. And of course, this would allow us to design better clinical trials for health span interventions. In other words, those last 3 points the ultimate goal would be to arrive at a state of being able to develop really personalized health span interventions for individuals. Okay, so why do we think we can do this? I think we can do this because we're at an unprecedented time to be able to systematically analyze heterogeneity, specifically at the cell level, at the single cell level and below. That is, we have really amazing single cell technologies on board in the world now, and the center has all of these available. There's heterogeneity beyond the single cell, down at the organelle level, even at the molecular level, epigenetics, transcription, et cetera. So we really have an amazing ability to analyze this now to where we can start applying these technologies to aging scenarios to understand the heterogeneity of aging. And ultimately, if we can understand how single cells and intracellular processes are affected with age, we should start to be able to model tissue biology, interactions between cells, and understand why different tissues and organs age, and ultimately utilizing this information to hopefully get an understanding of how organismal aging is heterogeneous. So to that end, the San Diego Nathan Schock Center has created 3 research resource cores to analyze the heterogeneity of aging with a focus on human aging. So the first core is the human cell models of aging core. The second research core is the heterogeneity of aging core. And the third is the integrative models of aging core. And I'll go through each of those briefly just to give you a flavor of what we have available and how we can maybe help your own studies with understanding the heterogeneity of aging. Lastly, I'll talk about our research development core and some of the unique aspects of our center with regard to bringing people into the aging field. So first of all, the human cell models of aging core. Our overall premise here was to try to develop novel cellular models of human aging. And to that end, we wanted to take advantage of some technologies that were pioneered in Rusty Gage's lab here at Salk and elsewhere, where fibroblasts can be converted, directly converted, if you will, rather than going through an induced pluripotent stem cell stage, but rather directly converted into different cell types. And when you do this, many cell types maintain the original aging features of the originating fibroblast, whereas iPSC technologies tend to erase these. And so the idea was to be able to generate a cohort of aged people that have been annotated for biological indices of aging, including bioenergetics, various frailty measurements, et cetera, at UCSD. But from these patients, we're going to generate, we're going to get fibroblast and generate iPSCs from them and bank those and have those available to the aging research community. But importantly, we're developing the technologies to directly convert those into different cell types like neurons, endothelial cells, et cetera, to have a corresponding set of aged tissue, cell types to analyze. With those in hand, further down the line, we hope to be able to start making organoids out of these cell types to look at interactions between cells and to begin to model the tissue microenvironment and how aging affects that and how that impacts the heterogeneity of aging in tissues and organs. So that's the overall goal, and we are well into starting this process, but developing it as we speak in year 2 of this center. Of course, to analyze heterogeneity, you need many of the technologies that I'm sure you're all aware of, but we in the center have access to cutting-edge technologies for single-cell RNA and ATAC-seq, for example, proteomic and metabolomic analyses, really are pioneering many imaging modalities for single cells, using, for example, artificial intelligence mediated imaging modalities, et cetera. So we're really pushing the boundaries here to be able to analyze heterogeneity at the molecular and cell level. And so this core brings those technologies to the field for your utilization. Of course, these types of analyses generate a large amount of data. And so you need a lot of data infrastructure to store that data, and you need to be able to analyze it, but not only analyze it in general, but analyze it sort of with an eye on age related phenotypes and how to analyze aging data. And so we are developing new online tools new mathematical modeling approaches, and new ways of analyzing aging data that we think are going to be very powerful in the future in terms of analyzing the heterogeneity of aging. So we envisioned for the center sort of an intersection between these three cores, where we have novel human cell models, utilizing the heterogeneity core to analyze those cell models, and then using the integrative models of aging core to analyze the data. And through an iterative process here, we hope to get better at analyzing these types of data, being able to model heterogeneity in new ways, and really move the field forward in terms of being able to analyze age-related heterogeneity. But from your perspective, you should be able to plug into these cores individually as well. That is, we're planning on making available the fibroblast iPSCs and directly converted cells to you to analyze for your own aging studies. If you want to make them yourselves with your own aging cells, we can provide protocols, vectors, and advice for how to do this. In terms of the heterogeneity of aging core, your institute may not have access to all of the technologies we're talking about, so we can be a place you can come to analyze your aging experiments in this manner. Finally, you may already have your own aging data sets that you've been analyzing, but want to analyze them in a different way, and using our online tools and our mathematical modeling approaches to get more out of the data that you've already collected. So with that, I'll end with the Research Development Core. Obviously, all the Nathan Schock Centers have a Research Development Core with the goal of bringing new investigators into the field. We give 6 pilot grants through the center each year for use in utilizing our research resource cores. The next call for applications will be in September of this year. And this is really geared more towards junior investigators, although we do accept applications from new investigators coming into the field who are more senior. Coupled to that, we have training workshops through each of the cores throughout the year, and we have a large annual workshop where the grantees can visit, receive training, and actually see the research facilities and interact with the core directors at the center. And coupled with that, we have a mentoring program where we couple the grantees with an experienced mentor in aging biology. And all of these activities are coupled to an annual aging meeting in La Jolla called the La Jolla Aging Meeting, or LJAM, that's held at the Salk Institute that's been quite successful. And so we have 2 days in tandem where there's the aging meeting itself and then the workshop for the center. So with that, I'll just acknowledge some important people involved in the center. So first of all, the executive team of the center is myself as the director, Rusty Gage and Anthony Molina are head of the Human Cell Models of Aging Core, Martin Henser and Peter Adams are head of the Heterogeneity of Aging Core, Tanya Sharpie is the head of the Integrative Models of Aging Core, Jan Karlsader, who is director of the Saul Glenn Center for Biology of Aging Research, is also on the executive team, and Alessandro Sacco is the head of our Research Development Core at SVP. Obviously, I want to acknowledge the grant, the P30 through the National Institutes of Aging and the Division of Aging Biology for the opportunity to start this center and this exciting new area. I want to shout out to AFAR and the NSC Coordinating Center, who have been helpful in many aspects of setting up the center. And finally, we've amassed a really terrific external advisory panel of Rosalind Anderson, Mike Snyder, Nir Barzilai, John Sedevi. And I really highlighted Milena Hansen here because Milena, who is now going to the Buck Institute, was instrumental in many of the ideas for this grant for the Research Development Core development, served as the director of the Research Development Core for the first year, and has graciously stuck on as an external advisory panel member. And so she's just really had a sustained positive influence on the center. So I wanted to thank her. So with that, I will end and be happy to take any questions.